In this screencast, we're going to look at chromosomal abnormalities. So, sort of up till this point, we've been looking at um, uh, genetic disorders or genes and stuff like that. So, problems caused by mutations in genes, so that you had an allele or a different version of the gene that was responsible for the genetic disorder or the recessive phenotype, whatever. But you can also get genetic disorders and problems with an organism based on the fact that their chromosomes, their whole entire chromosome is duplicated or absent or messed up somehow. So these are kind of like large scale, oh, it says right there, large scale chromosomal issues. And so some of these can cause genetic disorders. Okay. Most of these end up leading to spontaneous abortion or miscarriage. Okay, so generally when you have a miscarriage, there's usually a reason for it. There's something wrong with the developing embryo. However, this is mostly in animals, okay, humans and other mammals. In plants tolerate a lot of genetic changes way better than animals do. They have all kinds of wackiness that goes on in their genomes. So let's look at some of the wackiness that can ensue. So the first thing we're going to look at is nondisjunction. Okay, and if you look at this as a double negative, so disjunction means to break apart, non means to not do that. So you do not break apart or you do not disjoin. And what this means is that when you have your pairs of homologous chromosomes at the metaphase 1 plate in meiosis 1, basically they do not separate during meiosis, during anaphase 1 of meiosis 1, as they are supposed to, which is when our alleles segregate because we separate our homologous pairs. So basically what happens is you have your pairs of chromosomes, bloop, 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 and rather than, kind of going over to, one to each side, they both go to one side and no chromosomes go to the other side. Okay, so one gamete receives 2 of the same chromosome and the other gamete receives none of that chromosome. Okay, and this can happen in either anaphase. It can happen in meiosis 1 or in meiosis 2. You can have a non-disjunction of homologous chromosomes, or you can just have a non-disjunction of sister chromatids. Okay, but anyway, the result is that you have some gametes with more than the usual number of chromosomes and gametes with less than the usual number of chromosomes. So if the non-disjunction happens in meiosis 1, you'll have 2 gametes that have extra chromosomes and 2 gametes that have fewer chromosomes. If the non-disjunction happens in meiosis 2, you'll have only 1 gamete that has extra chromosomes, another gamete that has too many chromosomes, and actually 2 normal gametes. The resulting condition, if you fertilize one of these gametes where non-disjunction has occurred, is called aneuploidy. And aneuploidy means like an abnormal number of chromosomes. Okay, so it's not diploid, it's not haploid, it's called aneuploid. It can either be 1 more or 1 less. Okay, and if you fertilize one of these gametes that has extra chromosomes or too few chromosomes, they will have an abnormal number of a particular chromosome. If you only have 1 copy of a particular chromosome, it's called monosomic. Monosomic is 1 copy. Trisomic would be 3 copies. Okay. If you have non-disjunction occur kind of more than once or with more than 1 chromosome pair, what you can get is polyploidy. Okay, and so you basically get non-disjunction of like the entire genome. So the organism will have more than 2 sets of chromosomes. So you can have 3 sets of chromosomes and be triploid. You can have 4 sets of chromosomes and be tetraploid. Okay, again, polyploidy is very common in those wacky plants, but extremely rare in animals. It does happen in animals, but it's extremely rare. Okay, and polyploids tend to be way more normal in appearance than aneuploids. Okay, so that's the way you can alter chromosome number. You can also alter the structure of a chromosome. Okay, and most of this has to do with breakage of chromosome. At some point during replication, mitosis, meiosis, whatever, chromosomes can break, unfortunately. Okay, and there are 4 types of changes that result from breaking of chromosomes. One is you can have a deletion, which removes a whole segment of a chromosome. 2, you can have a duplication, which would repeat a segment of the chromosome. 3, you can have an inversion, which basically like flips it, so it's backwards. Okay, it reverses the orientation of a piece of the chromosome. Or 4, you can have a translocation, which moves a segment from one chromosome to another chromosome. Okay, and this is what Owen has. All right, and this just shows you kind of how they are. So in a deletion, this segment of the chromosome is removed. So you go, you see in the resulting chromosome, there's a piece missing. A duplication, okay, would take a segment of the chromosome and repeat it. So the resulting chromosome has kind of extra DNA or extra copies of that section. An inversion would take a section of chromosome and kind of flip-flop it. Okay, so instead of this section going genes B, C, D, it would flip that orientation and go D, C, B. So it's the same stuff, but it's reversed. Okay, and then a translocation usually involves 2 chromosomes. Okay, so it would take this chunk, and they swap places. So this piece goes onto this chromosome, and this piece goes onto that chromosome, and you get kind of big jumbling of genetic information. And this can be, can have no effect, it can have a huge effect, or it can have a little effect. Okay, so now let's look at some of the, so those are the types of chromosomal abnormalities or alterations that we're going to be talking about. Let's look at some human disorders that are a result of this type of thing. Okay, and alterations of chromosome number can be associated with some very serious disorders. And some types of aneuploidy upset the genetic balance a little less than others, resulting in individuals surviving to birth and actually leading kind of lives that are of about normal length. Okay, but individuals who do survive have a set of symptoms or a syndrome that is characteristic of whatever type of aneuploidy it has. Kind of the most common aneuploidy, the one we're most aware of, is Down syndrome. Okay, Down syndrome is trisomy 21. So it's an aneuploid condition that results from 3 copies of chromosome 21. So you would have a gamete that had nondisjunction at chromosome 21, so it has 2 copies, and it would fuse with a gamete that has normal, has 1 copy, and 2 plus 1 equals 3. Okay, so Down syndrome occurs actually relatively common if you think about it. 1 out of every 700 children born in the United States has this condition. Okay, the frequency of Down syndrome increases with the age of the mother. And again, it might have to do with the fact that the eggs are getting a little bit old. And so the nondisjunction is much more common. Okay, and you can see here if you look at, so this is kind of the phenotype of Down syndrome that we're familiar with. And then if you look at the genotype, there are 3 copies of chromosome 21. Okay, and this is the little girl because she's XX. Okay, if you have aneuploidy of sex chromosomes, nondisjunction of sex chromosomes also produces a variety of conditions. There's Klinefelter syndrome, okay, is an extra X chromosome in a male. So you get the phenotype XXY. Another type of condition from nondisjunction is monosomy X, whereas this is called Turner syndrome, and the females are XO. They only have one copy of the X chromosome. Okay, and some disorders caused by some structurally altered chromosomes, so the deletion, the inversion, the translocation, that kind of stuff. There's a syndrome called Cri-du-chat, which is cry of the cat, and we'll look at a little video in class about Cri-du-chat. But Cri-du-chat is a result of a deletion on chromosome 5. Children born with the syndrome are mentally retarded. They have lower than average intelligence and has a cry like the sound of a cat. And individuals usually die when they are very, very young. There are some cancers, chronic myelogenous leukemia, that's caused by a translocation. And this is, if you have a look at this, so again, this is the CML translocation. And so again, the end of chromosome 9 and the end of chromosome 22 switch places. Okay, and you get translocated chromosome 9, and then this translocated chromosome 22, and it's the chromosome 22 that is the problem that causes the leukemia. Okay, so that was a look at some disorders that occur from very large issues with your DNA rather than just a mutation that causes an allele. Okay, and that's it for now.